And it's my pleasure, very much my pleasure, to introduce Louis Lefebvre and his talk this afternoon. And as someone who works with very simple organisms, I always wonder what it would look like if a frog could just make up something that was very adaptive for it and do it, and i.e. if it could innovate. So I'm very jealous of people who study those kinds of animals, being now mostly birds and mostly primates, but some really smart fish like cichlids are known to do some innovation. So Louis is really one of the pioneers in the field of animal innovation, and I always think that parallel to that, he's really at the forefront of doing real integrative animal behavior, which in my mind is addressing all four of Tinbergen's questions: survivorship, evolutionary pattern, mechanisms, and acquisition, and putting all of those questions together to give us a really deep understanding as to why animals behave the way they do, which is really the single question that Tinbergen ends that paper with. He says, well, we can ask these four questions, but we're all interested in one thing: why do animals do what they do? So I think today we'll hear about why do animals innovate when they have that capacity.
C'est drôle de parler anglais à un auditoire qui est probablement majoritairement. Go.
This evening, the talks are not here, and the talk is not here. It's in a different place, which is in your schedule, which is in your book, and you should remember to bring your name tag, your badge, and it's not at 7:30, but it's at 7 p.m. So I think I got the 3 points. Okay.
Okay, so what I've been trying to do over the past several years is to look at cognition in the wild, and as this sentence says, try to operationalize it. So the word operationalize means define it in the way that we can measure it quantitatively, which means that certain aspects of sentience might not be amenable to study with the system I work with. So I want to warn you right in advance that very little of this will be amenable to issues of, how should we call this, very, very complex cognition. So in order to do that, what I think we need to do, and other people do other work which is really interesting and fine, that's just the way I feel my group needs to work. So in order to operationalize cognition in the field, you should have a definition of it that makes it quantifiable. That is, you should be able to tell that species A has, say, 37, and species B has 22, and species C has 0. So there's a quantification aspect to it. But also, you should be able to manipulate it experimentally, both in the field and in captivity. I also want something that is sufficiently simple that we can measure it in a very, very broad array of species. For instance, a study came out a while back, a couple of weeks ago, by Nathan Emery's group, in which they look at rules for reversal learning in parrots. And they conclude that parrots have an ability similar to that of great apes to generate reversal learning rules. If this is something that only great apes and parrots do, then we're stuck because we have the tip of the iceberg of cognition. And I'm interested in all the ice that's below the water level, as well as the tip of the iceberg. And that's sort of the philosophy. If you want to understand the tip of the iceberg, you have to understand everything that's underwater. So in order to have a simple definition, then that allows you to have a very broad range of taxa instead of only certain select groups. And again, I'm not saying anything against people that work with very specific groups. I'm just saying this is the way we work. And also, if you want an integrative approach, then you have to speak to people in different fields. You have to speak to people in neuroscience, in psychology, in ecology, and evolution. And if your concepts are taken only from psychology, then people in ecology won't understand what you're talking about, and vice versa. So you need to be careful of the way you define your behaviors and the kinds of concepts to use. And again, I'm not saying everyone has to do that. That's just what we do. So here I'm going to start with the same point that Simon started with yesterday, which is the first innovation that was described in the literature in 1921, which is the opening of milk bottles by tits. And what was peculiar about this is that it was a new food, and they were only drinking the cream because birds don't have the enzymes to digest the carbohydrates of milk, so they're just digesting the lipids. It was a new technique. Some people say it's not new because this is how they search for insects, by removing bark. So people disagree on whether the technique is new, but in any case, at least the food is new. The place is new because they are on suburban doorsteps. And English people being so nice, there's very little predation risk on an English doorstep. Perhaps in other countries, the tits would have been shot. I won't name that country. There's also a new time because it was noticed for the first time in a small town in southern England in 1921. So what you have over here is an innovation which is defined by opportunism, which means you take advantage of something that suddenly appears. And there's neophobia that has to be low because you're approaching something completely new that could eventually be risky, which is the bottles and the doorstep. And there's also boldness and tameness. Despite the fact that these people are English and they're very sweet, it could still be risky to have the milkman or the residents near you when you're opening the milk bottles. And for several decades, the thing that people asked about this kind of behavior is, how did it transmit? They didn't ask, how did it start? They asked whether or not the behavior could be transmitted, because what the initial paper in 1949 by Fisher and Hine showed is that the behavior, as Simon showed yesterday, was seen in more and more places over the map of the UK. And therefore, the question was, how did it get to transmit? So what's very nice, and Simon mentioned the very nice work of Lucy Aplin and others in Oxford, they developed experimental proxies for social learning, in which they developed tasks of obstacle removal. In the Aplin case, it's the left to right pushing of the door. In the case of other animals, it's always removing something similar to the cap of the milk bottle, ripping, lifting, whatever. And they also developed many controls for alternatives. Simon showed you a list of about 72 possible mechanisms for social learning. And so people in the field of social learning are always worried, is it true imitation or is it something simpler? So they have all sorts of controls. So there's a strong research tradition here that we can use because of the controls and because of the obstacle removal tasks. What we do if we're going to study innovation and not social learning is we remove the trained tutor naive observer part of this thing. And only recently have people asked, well, if it's not social learning, how come it was tits that innovated? How come it wasn't emus? And you can tell just by looking at their eyes why it's not emus. En québécois, on dit un cavoir en webé, que so maybe cognition, maybe intelligence has something to do with it. Maybe. We'll see later on. So then you can ask whether there are other cases, and if there are other cases, you can start working with that. So I'll give you a series of examples of other cases. So collared doves have invaded Europe from the Mideast and through Turkey, and they've spread to different parts of Europe, and they're very urbanized and opportunistic. And in this town in Catalonia called Sitges, people have cat shelters. They feed and they give little houses to feral cats in the middle of the city. And in these little houses, the feral cats can give birth to kittens, and they're sort of sleeping there during the day, and they come out at night, and the people give them food. And what the collared dove is doing over here is it is going to the food bowl, which has pellets. So the dry pellets for the cats are being taken by the collared doves, and the collared doves are eating them. There are cats there, okay? So this is a risky thing, and this is the thing that collared doves do not do. So when we see this, we say, this is novel, this is a novel place, a novel behavior, etc. It's never been described. This is what we look for. Over here you have one step further in the use of these dry pellets, which is what grackles do, which is to take the dry pellets and to dunk them in water to soften them, and that makes them edible much more quickly. And in some cases, you have individuals that steal the dunked pellet of the individual. So a few seconds before this photograph, this guy had a pellet in its beak, and you can see the opening, and you can see it going, what the happened? And this guy just stole it. So I'll show you what the behavior actually looks like. And it's a very short video. So it's going and it's dunked it in water. And this guy was approaching to steal, okay? So this is the kind of thing we see. And here is another innovation that I recently found in a journal. So what you have over here is an abandoned mine. And the abandoned mine has bats in it. And as the bats go in and out of the abandoned mine, there are crows that either stand on this thing over here or actually go inside the mine to get the bats. So again, what you'll see is a bat will fly out of this opening from the abandoned mine, and it will be caught by the crow that just grabbed it. And again, there's another kleptoparasite there that came in, and if it can grab it from the other one, it will. Here is another innovation that was noticed by Simon Reeder. And I guess he'll forgive me for saying it again. He was at the bar of the Colony Club Hotel when he saw this innovation. The bullfinches that have territories will steal sugar packets. These are nectarivorous birds. They're omnivores, but they feed on the nectar from flowers. And if sugar is given in open bowls, they'll eat sugar from the open bowls, but they're territorial. So one of the neat things of a territorial bird that's opportunistic is that they'll come down to the tables, they'll grab the sugar packet, then they fly off to their territory and eat the sugar as they're at their leisure. So this is what the behavior looks like again. So this individual is looking for a hole that's already been made, and there is no hole, but it's the mate of this individual that does the opening. So it's just basically looking for the hole. And there are other species like banana quits around who are incapable of piercing because of the shape of their beak. So they're basically looking for things that have already been open. Once in a while, innovations have characteristics that might create problems for people. So for instance, if you go to Rome on a sunny day, the Pope can be releasing white doves for peace or whatever. The problem is that there are now gulls and crows that kill the white doves released by the Pope as the Pope is releasing the doves, which is a very bad thing if you're a Pope. But I guess there are other perks to the job. So I assume that these poor children are crying as this is happening. So these are one of the things that you see as you do this. And I did see in Catalonia a gull kill a pigeon in a city square. And they are becoming predatory in places like that. So basically what we do is we go through as many ornithology journals as we can. Thousands and thousands and thousands and thousands of short notes. And up to now, we have over 3,800 cases in over 1,250 species out of the 10,000 species of birds, for which we have at least one innovation. And we validated this against many confounding variables, and we look at the taxonomic distribution. So which kind of bird has, say, 33, which kind of birds has 6, and which kind of bird has 0. And of course, there's a large measurement error at the species level, so we tend to work at higher levels where there's an averaging out of the measurement errors. Then what we can do is we can take this taxonomic distribution and use it just like any other phenotypic variable. And we can then test it against a whole series of other variables in ecology, evolution, life history theory, cognition, and neuroscience. We just measure. That's all we do. As we're going through these thousands of journals, we come once in a while with interesting, if at this particular case, it's not just interesting, it's sad. Okay, so I don't know how many of you hope that for once the coyote will win. Okay, the roadrunner is very innovative. For a cuckoo, it's very innovative. It's an omnivore. It does lots of interesting things. But in the cartoon, it always wins. But in real life, Wile E. Coyote would be happy to have read this paper. And I think it's in Austin, Texas, that this was seen. This is from the Bulletin of the Texas Ornithological Society. And what they have seen is this poor loser roadrunner. So they saw this roadrunner. There was a tennis ball, okay? So the poor roadrunner, who had been defending a territory for 6 years against no other roadrunner, never attracting a mate in those 6 years, and routinely offering food only to its reflection in a window, decided that its life was just too depressing, and it was at last going to have a baby roadrunner. But there were no eggs, right? So it found a tennis ball. It built a nest around the tennis ball. It incubated the tennis ball for 2 months. And this is such a sad story, right? But you find these very interesting stories as you're going through the thousands and thousands of cases. So basically what we have to work with is a count. We've got thousands of cases in hundreds of species, and we look at the distribution. And we correct the distribution by several confounding variables. The one we use most often is research effort, because you would predict that if a species is studied more intensively, it's going to, just by chance, be seen doing more novel things. And we look for keywords like the word new, novel, never seen before, opportunism. And our approach is black box in the sense that I'm measuring something and I'm not quite sure what's in that. It could be things that's very simple. It could be things that are very complicated. We don't know. A priori, we take everything. As soon as an ornithologist says, I've never seen this before, this has never been reported, this is new, and therefore we are using deliberately broad criteria, and we're targeting things that are not just intelligence, that are opportunism, that are also generalism. And because of this, we are not making any a priori choices. We are taking everything, which means that we can test ideas post hoc. We can take the thousands of innovations and say, okay, some of them are technical, some of them are new foods. We can do the same with tool use. We can say, okay, people say that a true tool is when you take something from the environment and you do this with the food. If you're taking the food and you're dropping it on something hard, that's not a true tool, it's a proto-tool. But if you're taking the rock and banging it on what you want to open, that's a true tool. People say this is more complex. It requires maybe more planning, whatever. We don't know what's in the box, but what we can say is, let's take all the true tools where you're manipulating an object from the environment and directing it to food, and let's take all the proto-tools where you're manipulating the food and doing something with the environment, for instance, dunking in water or dropping on a rock like a gull will do, and we'll see whether the size of the brain or the part of the brain that we think is involved, like the nidopallium, is it larger in true tool users because true tool use is supposed to be more complex. So a priori, we take everything, but post hoc, we can sort of clean the database and test for ideas. So what you end up with is something like this. You end up with a taxonomic distribution of innovation rate, and innovation rate is the number of innovations we see corrected for the most important confounding variable, which is usually research effort. So if high innovation rates, which is this side of the line, are occurring in animal groups, avian groups, that are all bunched together, then it means that it evolved potentially once in the ancestor. If it's present in different groups that are separated phylogenetically, it means we have repeated independent evolution of, and Simon showed a graph similar to that yesterday, in which the great apes, the baboons, the macaques, the capuchins, all were independently evolving high rates of what the figure he showed was general G, the general process intelligence. But you could say the same thing for innovation. So this is an old phylogeny of a few years back. The annoying thing with birds is that the phylogeny, the molecular phylogeny, changes every 5 years. So you have to redo everything. So for instance, what you see here, the falcons and the eagles and the buzzards are now very distant from each other. The psittaciforms, the parrots, are now considered to be much closer to the passeriforms, the songbirds. But the whole thing has to be redone. So this is an old phylogeny because it was the best illustration of the type of graph you can do. So you end up with things like, oh, well, who are the most innovative birds? Well, clearly the corvida, the crow-like birds, are the most innovative. So you end up being in the news and people saying, oh, crows and jays have topped. So there's a journalist that was hired by NSERC to do a little blurb on this research, and he used the word bird IQ. And that made it to the newspapers because of the word IQ. I never said I'm measuring IQ, but this guy said I was measuring IQ. And it's true that the corvids, using other methods. So today, this paper just came out. If you're looking at your news feeds for interesting cognition, they just showed that a New Caledonian crow is capable of building a paper tool of the correct size to fit into a vending machine that gives it food. And to remember this from the trials in which it was trained. So there's only a certain size of paper that works in the vending machine, and it remembers, and the day after it produces the right sized tool. Sometimes you get into trouble. So they made me say, okay, crows are the most intelligent. Who's the dumbest bird? I said emu. And people in Australia were very upset because emus are the native, they are on the nation's coat of arms. And say this person from the Center for Neuroscience at the University of New England in Australia said emus have been defamed. So they're not happy. What can you do? Okay, so I can be shot by Emu Liberation Front. Okay, so then what you can do with this is use your taxonomic distribution of innovation rate to test with correlational methods that take into account many confounding variables and take into account phylogeny. All of these particular ideas that can be tested. And so the techniques that are used are, we used to use independent contrasts and general linear models, phylogenetic generalized least squares regressions, phylogenetic generalized least squares, et cetera, are used by people that I work with, including Daniel Saul at the Autonomous University of Barcelona. And what you end up getting is something like this. So here you have the innovation rate measured with fairly complicated metrics. And these are very complicated models. And so what you get is the relationship, say, between innovation frequency in this case, depending on, I think these are avian orders, and invasion potential, which is the ability to survive after you've been introduced into a new country. So for instance, you take birds from England, you bring them to North America, like we have starlings in North America, we have house sparrows in North America. They do the same thing in Australia, then they bring these birds to New Zealand. In most cases, the starlings and the sparrows will make it when you bring them to a new country. So what you then do is to look at what was their innovation rate in the country of origin? What was their innovation rate in the UK? And what you find is that the species that have high innovation rates in their country of origin can survive an introduction much better worldwide than species that have low or no innovations. You can get actually species that are introduced, like pheasants. So people introduce pheasants to countries where there are no pheasants because then they want to shoot the pheasants. The problem with this is that the pheasants die before you get to shoot them because they're not good colonizers. So the kinds of things you can then test with this, and for purposes of time, I won't give many details, but all of these studies, for instance, so innovation rate is associated with colonization success after an introduction. It's associated with habitat breadth, with diet breadth. Migratory birds tend to have fewer innovations in their country of origin. So resident birds, and obviously you need to take into account the fact that migratory birds are only there for half the year. So that's taken into account. And also Simon mentioned yesterday, it's the same result in primates. There's a cost of acquiring pathogens to being innovative. So innovators tend to try out lots of things, and they acquire pathogens as they're doing this. So the main cost that we've been able to see, in addition to life history costs, is the cost of acquiring pathogens and different kinds of pathogens. In terms of life history, innovative taxa tend to have longer lifespans. They tend to fledge later on. There's no relationship with the slow-fast continuum. And species with low brood value tend to be more innovative. Low breed value means that you're not putting all your eggs in the same basket of the next reproduction. You can skip a year, or you can skip a season, or you're producing one offspring or two offspring. Other species that produce 10 offspring in the first year, which is high brood value, tend to be less innovative. So for instance, ducks that produce lots of babies in one shot tend to be less innovative. There's also a relationship between innovation rate at high order levels and the number of species, and also the number of subspecies. So there's an idea out there that's called behavioral drive that says that if you acquire novel behaviors, you can move into new niches. And by moving into new niches, by having flexible behavior, that then can lead to speciation, divergence, and acceleration of evolutionary rates. And there's support for that with innovation. In terms of cognition, there's a positive relationship between innovation rate and tool use, but we were not able to find one with social learning in birds, contrary to what Simon finds with primates. And finally, when you look at large data sets of bird brains, the large data sets, we have, there are data available for almost 2,000 species of birds in terms of brain size. There are data available for about 66 species in terms of parts of the brain, like the pallium. There's a positive relationship. And one of the interesting things is that people say, wow, when a macaque washes food, this must be really, really intelligent. So these are very famous cases from the 1950s in which Japanese macaques were seen to wash different kinds of foods, in this case, potatoes. But crows will also do that. So in this case, you have people putting out pieces of meat on bird tables for crows. And the pieces of meat start attracting insects and attracting ants. And so what the crows do is they take the piece of meat, they go wash it in water to remove the ants, and then they have the piece of meat without the ants. This is not very different from what the macaques are doing, except that people are not interested in how it spreads, and it doesn't spread very much beyond the individuals that are doing it. What this indicates, however, is that when you look at innovativeness, there is strong indication of convergent evolution of innovativeness in birds and primates, despite the fact that there's over 300 million years of separate evolution of birds and primates. And we could multiply perhaps these number of cases of convergent evolution if we were able to get good databases. For birds, we have several thousand cases. For primates, Simon has 600 or a little bit more. So it would be nice to do it with other taxa, But when we tried to do cetaceans, when we tried to do cephalopods, the databases are too small to run comparative analyses like this. So we were able, of course, cetaceans are said to innovate. But if you find 25 cases in all the species of cetaceans, you can't do anything with that because you want to be quantitative. Okay, so these are all different aspects in which primates and birds show very similar patterns. So this is a figure from a paper that we wrote together, Simon and I, with Daniel Saw. And what you can see here, the data, the details are not important. The point is that birds and primates, the birds on the left, primates on the right, show very similar trends for several types of measures. So that's evidence for convergent evolution. If you go back to the sugar packet opening case, these birds are extraordinary. They're very, very easy to work with. They're very tame, and they happen to be in Barbados, where it's plus 28 degrees in winter instead of minus 28. So it is very nice and very easy to do research at McGill's Field Station. And these finches are so tame that you can get them to do obstacle removal experiments in the wild without catching them, okay? This is an example of an obstacle removal experiment done in the wild. So what this individual does is it first says, okay, I'll try the lid. Maybe not the lid. I'll just push it, and there's a drawer. And it uses the drawer opening by pushing. So there's different ways of solving this, and these birds will do this very, very readily. So they're easy to work with. And you can make things a bit more complicated. So in this case, they habituate very well to captivity. In this case, you have to pull out this thing, and then you have to open the lid. This is something that they can do quite readily. So what you can then do is ask, okay, once we have this experimental proxy, we can again ask questions about ecology, evolution, cognition, and neuroscience. So I'll be giving a little bit more detail here because this crowd is probably more interested in this aspect than in the large database comparative approach. So for instance, we know that urbanized bullfinches are better at this than rural bullfinches. So there is an effect of urbanization. There's an effect of social competition. When competition is too intense, this reduces the ability to innovate. You can show that there's a relationship between reproductive success and the ability to solve obstacle removal problems. The work hasn't been done on these bullfinches. The work has been done by the ladies sitting over there. Larcochard on tits in Sweden. Other work has been done on tits in England, where they look at whether individuals that are better at solving obstacle removal problems have a higher reproductive success. And one of the things that Law was able to show is that the rate at which they're provisioning the offspring is also associated with the ability to solve obstacle removal problems and fledge more offspring. So what this means is that there could be selection on behaviors like this. Finally, what I'll be talking about a little bit more in detail is that you can go deeper into the cognitive mechanisms behind these behaviors, as well as what's going on in the brain. So we're going to go beyond brain size, looking at the actual neurotransmitters and the receptors of these neurotransmitters. So the question for a crowd like this is, if you're going to measure cognition, what ecological hypothesis should you be basing your predictions on? So we know that there's lots of work done on spatial cognition. So if you're hiding food, if you're a brood parasite, if you migrate, you might have a different use of space than if you don't do any of these things. If you're social versus non-social, Simon asked the question yesterday, is there something about social learning that is adapted to social animals? He answered no, but that could be a possibility. What we're looking at, though, is more opportunistic generalism. So we're not looking at social specialization or spatial specialization, but opportunism generalism. So If you're an opportunistic generalist, you need flexibility. And if you're looking for extractive foraging, there's a tradition in social learning and innovation, because innovation started in the field of social learning, to use the obstacle removal task that people have been working on based on what the tits were doing with the milk bottles. So how should we measure flexibility? So there's a school of thought that says we should measure flexibility by looking at obstacle removal. We give them a closed apparatus with food in it, and this is the way we need. And the reason is traditional. It's because people have been doing this for a long time. There's another school of thought that says, no, you need to look at reversal learning, because in psychology, the main task that's used for flexibility is reversal learning. So there's basically two schools of thought here. And the question is, should you be using this? Or maybe you should be using both. And if you use both, do you get the same result? That's the difficulty, because basically, you don't get the same result. So when you measure obstacle removal, in this case, the task is, so this is the work of Holekamp's group on hyenas, but she also worked with Benson Aram, and they looked at dozens of species of carnivores. And in all cases, it's food hidden inside a box, and you have to find a way to open the box. This is the task that they use on the chickadees in Pravosudov's lab. You have to remove this block over here to get the food inside the tube. Here, this is a pure reversal learning, discrimination learning task, because there's nothing to remove. So what you have here is seed that's edible because it can be removed, and you learn that it's in the green box. And here it's exactly the same visually, but the seed is glued to the bottom of the Petri dish. And this task was invented by Jean-Nicolas Audet. And so here it's visually identical, but if you try to peck at this seed, you can't remove the seed from the dish. So you learn that the yellow is no good, and then you measure how long it takes for them to learn yellow's no good and green is good, and then you reverse, you just switch. And the next time, and then you try to see whether or not one species or one population or one individual is better or worse than the other at learning the discrimination and then reversing the discrimination. This is flexibility. However, you need to ask yourself, how often does this exist in the world? How often does the world change completely from one day to the next? Bumblebees maybe encounter flowers that for 4 days give you a very high rate of nectar production because the flowers smell like jasmine and they're blue. And then 3 days later, the blue jasmine smelling flowers are no good, and it's the yellow whatever smelling flowers that are good. So reversal learning, you didn't go to the yellow, you went to the blue. And 3 days later, you don't go to the yellow anymore, you go to the blue. So bumblebees encounter reversal learning, but who else encounters reversal learning? Imagine that suddenly the coffee machine was in the toilet, and the toilet was where the coffee machine was. Are we predisposed to cope with reversals of this type? No, but psychologists insist that this is the way we should measure flexibility. So I'm not sure that ecologically it makes any sense. And you can pretty much tell that we prefer obstacle removal to reversal learning, if only because the results are completely different. So all of the cases for the moment in which people have done both obstacle removal and reversal learning, you end up with either a negative relationship across individuals, across populations, across species, or a zero relationship. So I'll show you what this looks like. So this is the paper that just came out by the Nowicki Searcy group. And they used obstacle removal over here, novel foraging. And there's no relationship, it's even slightly negative, between performance on novel foraging and the color association and the color reversal. When we look at urban versus rural bullfinches, again, we get a difference in how good they can solve the problems that you saw, opening an apparatus or opening the tunnel. The tunnel is where they have to pull the little popsicle stick in order to get the thing to open. So there's a difference. The rural ones have a higher latency. They take longer to learn, and the urban ones learn faster. But there's no difference in terms of either their discrimination or their reversal learning. When you look at the interspecific level, you take your bullfinch, which is extremely opportunistic and innovative, and then there's this little species called a black-faced grassquit, which is very close to bullfinches in phylogenetic terms, but basically it's a seed specialist that just forages in the grass in Barbados in the same environment. And when you bring them into captivity, they are hugely different in terms of solving obstacle removal tasks. In fact, the task we gave the bullfinches, which they solved fairly fast, none of the 15 birds we tested in the other species was able to solve it, so they just can't cope with it. When you give them a detour reaching task, which measures inhibitory control, again, your opportunistic innovative species does well, and the other one does more poorly. But there's no difference in their discrimination or reversal learning. So there's a mystery there. Maybe the mystery can be explained in part by a speed accuracy trade-off. So what the speed accuracy trade-off says that maybe if you try lots of things really fast, you'll be able to solve an obstacle removal problem. But if you try lots of things really fast, you're going to make mistakes in discrimination learning and reversal learning because you're going to try everything. So trying everything really fast makes you a good innovator, a good solver of obstacle removal tasks, but it makes you poor at reversal and discrimination learning because you have to sort of hold back and just choose the correct one. You just choose one of the two. If you choose quickly the two, you're out. It's a mistake. So maybe there's something there. So you can then ask what kinds of behaviors predict innovative problem solving. So at the level of individuals, obviously the ones that are more exploratory, the ones that are less neophobic, the ones that don't fear novel objects as much, but also the ones that respond well to feedback from the environment. So in this particular task, the food is over here, and there's a transparent lid. And if you're pecking near the food to get at the food when you're hungry, it's like an obstacle, it's like a detour reaching task. Basically, if you're pecking at where the food is visible, you won't get it. You have to peck at the sides in order to remove the lid. You can't remove the lid by pecking directly at the center where you see the food. So the ones that are directed towards the food, and they all try that at the start. So at a certain point in time, the ones that are going to solve this are the ones that stop doing this and then try other things and then realize that if you peck over here, it moves. And then you can solve it. In hyenas, the Holocomp group has also found that the more persistent you are, the less neophobic you are, and the greater the motor diversity of the things you're trying. This is what leads to solving this problem. So if you only try one thing, or if you hesitate in touching the novel apparatus, all of that is going to make you less capable. And again, it might be some kind of a speed accuracy trade-off. You're trying everything, and that's how you get to the novel food. But if you were measuring discrimination and reversal learning, that would be a problem because you're trying everything. So what kind of things do we expect would be occurring in the brain that could correlate or cause innovation? So by correlate, I mean if we're looking at innovation rate across taxa, and then we're measuring brain size across taxa, or size of the equivalent of the cortex across taxa, this is correlational. It doesn't tell you anything about the control. In order to control behavior, You have to interfere directly with the synaptic events. You have to block the neurotransmitters or the receptors, or you have to modify the action of the individual neurons. So this is where the causal knowledge about what controls innovation needs to eventually be directed. So when you look at a bird brain isn't made like a mammal brain. There is no cortex. So there's no series of layers on the outside. And things are pretty much reversed. So in a bird brain, you've got nuclei in the middle instead of things on the outside, instead of layers on the outside. And the functional equivalent of the prefrontal cortex is at the back. It's over here. It's called the nidopallium caudolaterale. It's NCL. So these are the things you want to measure in birds if you want to measure the equivalent of the associative non-visual cortex of mammals. And when we look at the size of the brain, people say, oh, but how can you be interested in size of the brain because the brain does so many things and it's a really stupid thing to measure. Well, actually, variation between taxa in brain size is almost all driven by variation in the nidopallium and the mesopallium. So basically, when a brain evolves to a bigger size respect to the body, it's because the nidopallium and the mesopallium. And someone sitting over here has a theory about why this should be in development. Barbara Finley, hello, Barbara. Ask Barbara why that part of the brain is the part that increases. Okay, so basically when you're measuring the size of the brain respect to the body, you're measuring the equivalent of the size of the pallium, nidopallium, mesopallium, with respect to the brainstem. Because the brainstem is basically uncorrelated with the size of the brain, it's correlated with the size of the body. What's highly correlated with the size of the total brain is the mesopallium and nidopallium. In addition to that, when you do the transcriptome of the entire brain, so this is work by Jean-Nicolas Audet. So he did the transcriptome of 6 different parts of the brain in the 2 species we talked about, the bullfinch and the conservative one, the black-faced grassquit. Notice here that the bullfinch, the opportunistic one, the innovative one is in blue, and the other species, the conservative one, is in red. There are fewer differences between species than there are between brain parts. And this is a principal components analysis of thousands of different RNA sequences. And what you find is that the entopallium, which is visual, has a very different transcriptome signature compared to the hippocampus. So people say the hippocampus is special. This supports the view that the hippocampus is special. But it also supports the view that nidopallium, mesopallium, and NCL, which is the caudolateral part equivalent to the prefrontal cortex, they function pretty much together. And this is what people call the associative pallium. So when you look at the size of the brain regressed against size of the body, there's a relationship with innovation rate. When you look at the size of the mesopallium, nidopallium regressed against the size of the brainstem, there's a positive relationship. People have been arguing that you should count neurons in the brain and not measure volume or mass. You should measure neuron numbers. So Susana Herculano-Huzel has developed a technique, which is basically you put brains in blenders and then you remove everything but the nuclei, and then you count the nuclei. And if you look at the data published on birds in 2016 by a group from the Czech Republic with Herculano-Huzel, it turns out that neuron numbers predict quite well innovation rate in birds, and that the relationship between the neuron numbers in the pallium regressed against the neuron numbers in the brainstem is equivalent to measuring the residual of the brain, the whole brain, against the body with a better slope than mass. So what this means is that to get a bigger brain, you get a bigger pallium. And to get a bigger pallium, you produce more neurons per cubic millimeter in those bigger palliums. So there's both an increase in mass and an increase in density. That's how you're getting a higher performing brain for this kind of thing. So we go back to the comparison of the two species. The bullfinch, which is innovative, and the grassquitch, which is very conservative. They're very closely related. They live in the same environment. And nicely enough, these are Darwin's finches. So you can put in your abstract and your paper, close relatives of Darwin's finches, which looks really good. And then remember the difference between, so there's no difference in discrimination learning, reversal learning, but there is one in innovativeness. And then we look at the types of receptors in the brain using 3 different techniques: the transcriptome, in situ hybridization, and immunohistochemistry. And this is again the work of Jean-Nicolas Audet and a group of people that collaborated. So when you look at all the receptors in the brain for glutamate, so there's many, many categories of glutamate receptors, what we find is that these 2 categories show no difference between our species. These categories, the NMDA and the metabotropic receptors, do show differences, but they show differences that are interesting in terms of what subtype of receptor there is. So, for instance, in the NMDA receptors, some of them are inhibitory and some of them are excitatory. So the ones that are upregulated in our innovative, opportunistic, problem-solving species are the excitatory receptors, the 2B, the 1, and the 3. The ones that are upregulated in the conservative species, the one that doesn't solve problems, are the ones that are inhibitory. And there's some cool research that was done by Zhou Qian on mice, in which they were able to show that if you have a genetically modified mouse that overexpresses the NMDA receptor 2B, it is better at solving a whole series of tasks. And they called it Doogie, like the TV show I never saw. I don't know what that mouse does, but apparently it's really bright. But if you overexpress 2A, which is inhibitory, those mice do poorly at the cognitive tasks. And this is something we also see in humans and other mammals, that the ratio of the 2B receptors to 2A receptors seems to be a key thing to measure in terms of the way the brain is able to cope with novelty. And this is exactly what we find in our 2 species of finches with the 3 different methods. So it's very robust, because here you have the transcriptome, here you have the immunohistochemistry, and here you have the in situ hybridization method that shows that it's this ratio that is important. And where in the brain is the ratio most expressed? The NCL, which is the equivalent of the prefrontal cortex. So we've got something which we think is showing that to become an innovator, you have more excitatory glutamate receptors, you have a higher density of neurons in your larger pallium, which leads to a larger brain. So you need all of these. And so I think I should eventually be closing. Mike, yes? No? Yeah. Okay. So what does this mean? Well, it means that if we find that the same neurotransmitter receptors, the 2A and 2B glutamate receptor, NMDA receptors, that are active and a key to human cognition and have been shown, once manipulated in mice, genetically modified mice. We have the same thing in birds. And it's the first case of evolutionary divergence, because what we find in mice is created in the lab. It's not something you find in nature. We've found it in nature. And what it means is that this is highly conserved, despite the fact that a bird brain and a mammal brain are extremely different anatomically. But it might be the same kind of neurotransmitter receptor ratio that's important. So what this means is that humans, if you want the take-home message here, We shouldn't just see our intelligence as something that is descended from the fact that we are great apes, but it also has something to do with the fact that we are much more opportunistic generalists than the other great apes. We are very good colonizers. We have basically colonized every part of Earth except Antarctica. And basically, we're very similar to crows. So convergent evolution is an important factor in understanding how our evolution has, how our intelligence has evolved. And some of my favorite, I'll skip this, some of my favorite animals in terms of animals that are very important for the evolution of innovation and intelligence, these are Homo erectus. These are 3 different ways of imagining Homo erectus. And honestly, if you look at peaks in the evolution of residual brain size in hominins, yes, there's an almost exponential and constant increase in brain size in hominins from the past 3 million years to recently, but body size varies enormously. So this is the body size graph logged. So this blue individual over here, this individual is 160 kilograms. Imagine meeting this guy, because presumably this would be something that is typical of a species with strong sexual dimorphism. Yet there's no dimorphism in the brain. So this was a huge guy that could beat up everyone, unless there was cultural transmission to give you access to a truck to beat up this guy. But there's hardly any difference in the brain. So there's no sexual dimorphism in the brain. The variance in brains over evolutionary time in hominins is almost constant. The variation in body size is very, very different from one period to the next. When you take those 2 into account, this peak, which represents the highest rate of increase in brain size, respect to body size, is in the Homo erectus period rather than the Homo sapiens. So hats off to Homo erectus, and that's what I have to say.
Any time for questions?
Yes.
I noticed in the video with, I think it was the bullfinch doing the tube pull, and it takes off the cap, and then there's a few moments where it looks like it's sort of casting about, like, what do I do with the cap? Yeah. And then drops it somewhere. I'm just wondering if you're looking for an observing different behaviors with the sort of the artifacts, the non-functional aspects of the apparatuses that you used, and so do they have these moments like, is this a useful thing? Is this not a useful thing? What do I do with this?
Yeah, this is something that you see when, with obstacle removal, they're working at the apparatus, they're working on the obstacle, and then when the obstacle goes off, they go, and this is what you saw. You saw it suddenly. And this is something also that you see when you try to understand how corvids, how crows deal with novelty. Crows in particular have this always neophobia, neophilia, neophobia. They're very careful at approaching something. They're very innovative, they're very exploratory, but at the same time, they're very skittish around novelty. And this is something that we see over here, this sort of surprise and jump.
And is there any sort of, do you observe any sort of split between species that sort of spend more time with sort of the side aspects of the apparatus and others that just go straight?
No, this is something we've done only with the Carib grackles. So the experiment I showed where it's attention to the movement of the apparatus and ability to move away from the obvious, which is equivalent to what people measure when they do detour reaching tasks, inhibitory control. So many people have found that inhibitory control is a good predictor of innovativeness and obstacle removal, except that this is not what they found in the hyenas. But again, there isn't 100% agreement within the literature. Some studies find, for instance, that all the tasks they give their animals correlate together, which indicates general process, G. Other studies find, whoop, no, there's this particular task. When we looked at bullfinches, we gave them a string pulling task, and we said, oh, we'll be able to see whether string pulling correlates with obstacle removal or with reversal learning. Nothing. String pulling correlates with nothing. I don't know.
Thanks.
Yeah.
Hello.
Yeah.
You said that innovation, is driven by the number of trials, the number of things they try.
Persistence and
how
motor
fast.
diversity.
But except for the pathogen risk, is
Yeah.
there other risks involved, accidents or things like that?
Curiosity kills the cat, which is a well-known thing. There are examples. Well, you find innovations, for instance, you find a dead bird because it tried to swallow something that's much too large. Or you find, for instance, there are, I think it's grackles that are able to ant with mothballs. So they take mothballs and they sort of put the mothballs on their feathers. And then there's one individual that actually thinks the mothball is edible, and it eats the mothball, and it looks really, really sick. So eating the mothball was not a good idea. There's a famous case of a Kia in New Zealand that died because it overdosed on chocolate. So it stole, Kias will steal things from your car and your picnic in New Zealand. So it stole a high cocoa content piece of chocolate, and it overdosed on because cocoa has a very stimulant property, and it overdosed on that. So yes, there are examples of non-adaptive innovations. There is a risk of death, but it's easier to measure the lice or the pathogens or the blood parasites on a population of several species than it is to chance on a bird that died because of an innovation it shouldn't have done. So yes, there's that risk.
Hi, I just have a quick question about something that might be a bit orthogonal to your talk. You mentioned there's skepticism about whether certain actions are true imitation. I found that kind of curious because some of the work I do, the claim is made, well, that's just imitation. So what were you referring to when you were, if you could just give me a sense of like what that debate is a little
Well, there was, I think at a certain point in time, people were talking about blind imitation and true imitation. So I don't know if this is still in the official table of possible types of social learning. But there was a time where people said, oh yeah, this is just blind imitations. You're just doing what the other person's doing without, quote, understanding what the other person's doing. That certainly has been, yeah. So again, it's like cultural transmission. When you look at cultural differences in the field, you need to demonstrate that the cultural differences in the field are not caused by something in the environment, that they're caused by almost arbitrary transmission differences. And again, this sort of doesn't correspond to what we know as culture, because Simon gave us examples of there's no real reason for people to speak differently here in Quebec or in France. But clearly, we need to be able, our ancestors that came here had to deal with cold better than the people had in France. So there are cultural differences that are adaptive. But the paradox is that when you're looking to demonstrate cultural differences in the wild, you have to demonstrate that they're not caused by the environment. And it's the same thing when you're transmitting novel behavior patterns in animals. You have to demonstrate that it's not the environment that's responsible for the behavior being picked up, but that it's the transmission independent of the environment. So again, it's paradoxical. Why should you learn something arbitrary that makes no sense? But if it makes sense, then maybe it's not purely social. So there's a possibility of a paradox here in social learning.
Okay, thanks.
Hi. I was simply wondering if you looked into differences between sexes, and if so, can you expect to see any differences between, female and male finches, for example? Or
In
anything
our
of that
work,
nature?
we've never found differences between sexes, even when we looked for them. When Simon looked at all the cases of social learning and innovativeness in primates, I think he did find differences between sexes and between age categories. But we've never done this for birds. There's just not enough data, and in our experiments, we've never encountered differences.
But there are differences in neurotransmitter ratios, or rather NMDA receptors, for example, ratios between sexes in
Yes.
certain areas of the brain. So
Yes.
you can expect then that you might see some differences in the behavior also.
Yes. Yes.
So it might be interesting to look at,
Right.
precisely because we don't have enough data, because we haven't been looking.
Exactly. Yeah. No, you're right there. Hi, Barbara.
Hi. So it's interesting trying to pull the different kinds of learning related to food gathering apart in some ways, and I suspect you've tried to look at different kinds of strategies. For example, you take an animal who's a sort of harvester, for a kind of animal that's been studied in optimal foraging kind of task, where you have a resource A, you deplete it, you go to resource B, then you go back to A. That seems a little bit reversal shifty to me.
It seems what?
So that seems like if there's a lot of animals whose food gathering is gather stuff from A till it's gone,
Yeah.
then go to B,
Yes.
then go to C, then go back to A because it's groaning in, which is a sort of reversal shift. And then you've also described the kind of thing where you're kind of just poking around as a food finding strategy, as opposed to doing this sequential thing. So I mean, my question is, do you find any sort of natural behavior in sort of kind of foraging that would map onto these things?
Well, basically, we looked at reversal learning and discrimination learning when we were looking for things in the brain. So because we were looking for the neurotransmitters and working in the brain, we figured there's this whole literature out there of neuropsychology and neuroscience that is based on tasks borrowed from psychology. So this is why we took tasks borrowed from psychology, like discrimination and reversal learning. And to our surprise, they were unrelated with the obstacle removal tasks. And even to our surprise, there was no relationship between the neurotransmitter receptor ratios differences. They are different, but they're not different. It doesn't correspond to a difference in discrimination or reversal learning. And this is something that goes against the literature, because most of the literature on the role of neurotransmitters in different parts of the bird brain, certainly if you look at the literature of the German group of Onur Günterken, who have worked a lot on the NCL, they would say the NCL has a role in reversal learning and discrimination learning. So how come you find a difference in the NCL of birds that have no difference in reversal or so basically, I would like to find the perfect task that I'll be able to link with the literature, but also that makes ecological sense. So I'm not sure it's reversal learning, and I'm not sure it's obstacle removal.
Well, the McLean one with the multiple species,
yeah,
might have that in it, because you could think, so the McLean study over a number of different species in particular problem-solving tasks was a detour task, and then A not B, and all of them involve having two possibilities of behavior in mind at once, and then discounting the bad one, when some animals just can't discount the one and they just keep doing, which sounds a lot like what you said.
But again, in the hyena study, they have the inhibitory control cannot predict obstacle removal. And it does in our case. It doesn't in the case of and in the case of the McLean paper. One problem with that paper is that it compares such a huge diversity of animals in terms of brain size that I'm a little bit perplexed by that paper because of that.
Hi.
Yes.
I was just wondering, when you conduct your studies comparing birds that live in the wild and birds that live in captivity, how do you process with the birds that live in the wild? Like, you capture them and then afterwards you put them back in the wild?
Yes. Yeah. We work mostly with the birds we catch. We keep them for a couple of weeks in aviaries, and then we release them, whether it's grackles, bullfinches, or grassquits.
The person who asked the question on that side just a moment ago is Professor Barbara Finley of Cornell, who was in this very same room in 2012, standing where you're standing, giving a talk that I actually never forgot about it. It keeps coming back to me, as a hypothesis about the origins of consciousness. And I'm pleased to announce that she'll be in the panel this afternoon at 4 o'clock.
Cool. Surprise guest. So those of you that are interested in how brains evolve to bigger size, Barbara has really interesting theories about this through a developmental model that takes into account the genesis of neurons in the embryonic stage and the point in embryonic development where you switch from multiplying progenitors to actually having these progenitors become neurons and start migrating to where they're going to be in the brain. This developmental process is at the base of evolution of large versus small brains, according to Barbara, and I'm generally sympathetic to that point of view. Thank you.
Is there a linear correlation between the brain size, the number of neurons, and the amount of connections between the neurons? Do they all vary?
See, when people say you should be measuring not the brain, but say the pallium, and then you should be measuring the number of neurons. Again, what you're saying is, well, maybe it isn't the number of neurons, maybe it's the number of connections. And the problem here is getting a sufficiently large database to be able to do comparative work. So we have over 1,500 bird brains in the literature, not me personally, 60-something pallium sizes. And as you saw, I think there's 28 species for neuron numbers. So if we were again to measure the number of connections per neuron, the problem would be, how do you get those data in sufficient numbers to be able to do a comparative analysis? So one of the excuses of working at the level of whole brain size was, this is what we have. Then Susanna Herculano-Husel came up with the method of counting neurons in large numbers of species. And again, large numbers, you're not getting into the hundreds, you're getting into the couple of dozens. So what do you do? And also the database of this group is heavily biased towards parrots. There's many, many parrot species. So to what extent is it useful to have, you know, a greater number of parrots, but also you have missing parts in the database? So again, it's a matter of, yes, you could make that prediction. How would you gather the database to test that prediction is another matter.
Because let's say for the same species, male and female, different head size.
Yes.
Do they have the same numbers? What varies? Because I guess a neuron is the same size for male and female.
Yes, there are larger brains in human males than human females, and if you correct by body weight or by body height, the residual brain size of males is either higher or lower than the residual brain size of females, depending on what you correct for. So there's no answer to that question. I don't know.
Okay, thank you.
Hi.
Yes.
This might be uselessly complicated the way I'll say it, but I think it's Simon Reeder yesterday that said that primates that are lower in the hierarchy are more likely to make these innovations.
Yes.
And that because they are lower in the hierarchy, these innovations won't stay, basically.
Yes.
I wonder if, related to the dominance hierarchy in birds, you see the same things.
We haven't looked at that. I don't know. For sure, every time we've tried to look at body condition, and you would presume that an individual that is higher up in the hierarchy would have a better body condition in the sense that the weight, taking into account the size, is larger in a dominant than it is in a subordinate. So there's body size, but there's also body condition. And we've never found any effect of body condition as a predictor of innovation, innovativeness.
Okay, and related to the species of birds that hide their food and go back to it after.
Yes.
Well, this is more of a fixed strategy. I guess the whole species do that. But do you think this has been initially some innovation that one individual has done for survival, and that species rely on that right now?
Possibly the origin of, how do you explain the origin of tool use and tool manufacture in New Caledonian crows? Presumably, there was an ancestor sometime within a population that only survived if they were able to get insect larvae inside crevices, and everyone else died. Then that would explain that in the experiments where you have juvenile New Caledonian crows that have never seen either tools or an adult using tools, they spontaneously start using tools. The same thing in Galapagos finches, the finches, the tool-using woodpecker finch, that has never used tools in a population that uses them very rarely. They start playing around with sticks. So this seems to be hardwired in them. And it's difficult to imagine that the origin of the hardwiring wasn't at some time related to differences between individuals, with some individuals innovating and then something happening that creates all or none selection. There's an example of Peter and Rosemary Grant in the Galapagos Islands. There's a species of finch. So in succeeding years, there were 2 years, I think, with lots of rain. So they didn't have to switch between dry season foraging and wet season foraging. And then there were 2 years of huge drought. And so every individual, every juvenile that didn't switch between the two types of foraging modes died. And all the juveniles that made it into the population the next year were individuals that were able to switch. So what this does, it indicates 100% all or none selection on flexibility. There's one known case of that, which is in the Galapagos Islands.
Okay, so related to what you just said, do you think that if food is abundant, innovation will be less likely to happen?
Probably.
Okay.
Probably. Curiosity kills the cat, and necessity is the mother of invention. Frank Zappa. Yes.
Est-ce que c'est correct si je pose ma question?
Vas-y, oui.
Okay.
Je vais traduire.
Moi, j'ai une question à propos du cycle de résolution de problèmes.
Oui.
Donc, quand, par exemple, les oiseaux, ils ont une tâche, comme nous, les humains, c'est un cycle qui est séquentiel et progressif, mais pour eux, ça va plutôt être par essai-erreur. Donc, je voulais savoir si vous avez trouvé un lien entre l'environnement socio-économique de l'environnement des oiseaux par rapport aux stratégies qu'ils développent pour résoudre le problème. Donc, est-ce qu'il y a un certain cycle, des tendances qui sont communes dans
So
vos
the question
recherches?
in English is there a difference between, say, populations or individuals in the way they approach a problem that has an obstacle removal? Is it, in some cases, just trial and error, trying everything, and in other cases, something rather different? To our great, maybe we should be measuring other things, but we measure latencies, and we measure whether or not an individual can do it. So only in the study in grackles have we measured how they do it. So in the bullfinch studies where we compared rural and urban, we did find a difference in success, but we didn't look at how they do it. Sorry.
Okay, thank you.
Could you tell me if I got that right? I remember that I think I remembered you said that basically conservative and innovative are seen like a dichotomy in the way you operationalize.
Yes, they're opposites. Yes.
Right. Okay. So therefore, my question is, you're also saying that your data shows that there's a strong correlation between the tendency to try lots of different things.
Yes.
Okay.
Yeah. Many studies, including the ones done by Andrea Griffin in Australia, suggest that motor diversity, and also the hyenas in Holocamp's lab, suggest that the more diverse the motor acts you direct towards the problem, the higher the probability of finding the correct solution.
Right. Okay. So we're dealing with consciousness and things like that in this thing, and it seems like innovation not only implies trying less of things, it also implies being able to realize that the new thing you just tried actually does work and sticking with it. And this even implies realizing that it's a better strategy than the previous one.
Yes.
And maybe it's just reinforcement or not, but deciding to stick with it, is that something you think?
Okay, so for instance, in the grackle study, we actually measured peck by peck, the pecks they directed at the apparatus. And there were pecks that we considered pecks that could lead to the finding of the food, which meant that they were directed to the sides, and pecks that were directed to the part where the food was visible at the center. So we asked, and so we actually plotted the curve of correct versus incorrect pecks as the animal was pecking. And we predicted that if there's such a thing as insight, as an aha moment, then you would have some kind of non-linear break in the curve. Okay, and so we measured about 30 individuals, and there was an indication of maybe a couple of them having a peck function towards correct and incorrect, which seemed to indicate a sudden change, as if they were sort of pecking at the visible part and then trying a couple of times elsewhere, and then realizing, aha, and then pecking only at the place on the side that produces. But this, we got this in only a couple of individuals, so overall we had to conclude that there was no evidence for anything sudden. And one of the definitions of insight is suddenness, right? The sort of aha moment where you understand, I have to stop pecking at where the food is visible and direct my pecks only at what's moving. But we didn't get that. We got individuals that seemed to be exploring different possibilities, but the rate at which they were producing more and more correct responses was increasing, but it was increasing linearly and not suddenly.
So no aha moment.
Well, it's very difficult to imagine what is aha for a grackle, but what could be aha in a response curve is a sudden change. And we got no sudden change.
All right, so one last question.
Yes.
Still, innovation implies that you stick with this new strategy. It's not like you try it, it works, and then you return back to what you were doing before. And to me, it seems like being conservative because you stick with the new strategy. So that's why I find it hard to understand why it would be a dichotomy.
No. But you do something new, but then you keep doing that new thing.
Right.
You don't keep changing. Presumably, the individuals, we only measure the time where the observer, the ornithologist, says, we've seen them do this for the first time. This is new. Presumably, if the ornithologist sticks with the species, they'll see this thing. The example I showed you where the crows were washing the meat, washing the ants off the meat. Presumably, they keep doing that. I can tell you that when you have grackles dunking pellets in puddles, we've done that thousands of times. Like, you see it over and over again. They do it.
So it's like they become conservative after a while.
Well,
because that's the whole point.
If innovation is created by opportunism, which means that either the definition of opportunism is that your normal feeding situation, you switch away from it either because it's not working and you need something else, or because you discover that this something else pays more than what you were looking for initially. So the dog pellets that are being dunked in water, softened in water, are something new, and they may be switching to that because they're hungry, or they may be switching to that simply because there's a good protein ratio in these foods. Because these are for dogs, so there's carbohydrates, but there's also, and there's fiber, but there's also more protein than in, it's equivalent to an insect, for instance. And it's all available there. So they learn to come to the place where we do the experiments. And they would keep coming and doing it. Yeah. All right, thank you.
Curious, of the, I forget how many it was, 1,250 or so species that
Yes,
innovations
with at least one, yeah.
Right. What sort of taxonomic distribution is that in terms of the groups of vertebrates or invertebrates? I don't know if there's any invertebrate examples.
There's 10, I'm not certain what your question is.
Oh, I'm interested in the distribution across, say, mammalian versus avian versus amphibians, say, or fishes. I know one example.
Again, we've got several thousand in birds. Simon has 600 in primates. Oh. When we looked at cetaceans with Lori Marino, when we looked at cephalopods with Graziano Fiorito in Naples, we could only get a few cases. People might have seen, you know, the octopus with the coconut shells in Indonesia. That's an innovation, but if you get one spectacular innovation of the Indonesian octopus creating a den by bringing together two halves of coconut shells, that's an innovation. Or the dolphins in Australia that use sponges to put sponges on their noses, basically to go into crevices. That's very nice, but you just can't, you need, if you're going to look at the taxonomic distribution, you need a sufficiently large data set that the numbers mean something.
Yeah, and that relates to the second part of my question. I don't know if Simon mentioned it yesterday. I may have missed that. But to what degree might the distribution that we know of be influenced by search effort or ability to observe the animal? Maybe hard, difficult habitat,
exactly,
maybe nocturnal.
Exactly. So some people say, well, have you looked at urbanization in terms of innovation rate? Shouldn't urban birds innovate more than rural birds? The problem is that the answer is yes, but that's because we can see the innovation because we're right next to it. So how do you remove observability? So we've looked at I think up to 14 possible confounding variables. We even went to an ornithology meeting and we asked ornithologists, if you saw this type of bird doing something completely new, how likely would you be to say, oh, and report it? And we asked if the bird is a crow, if the bird is a chicken, if the bird is a pheasant. And we put these numbers into multiple regressions. We tried everything. And right from the start, this method, I didn't expect it to work. I mean, the people say, you know, when we were getting rid of anecdotes in animal behavior. So if one anecdote is wrong, how come 3,800 is correct? It's just 3,800 times wrong. It's just, if you interpret the anecdote, that's the problem. I don't interpret them, I count them. So there's a difference. But even if you count them, how do I know that they're not affected by? So we've tried tons of confounding variables. Simon has put in confounding variables. Byrne and Whiten, in their deception catalog for primates, look at confounding variables. And every one of our studies has the confounding variables. And if at a certain point in time, someone says, aha, I found the confounding variable, and everything you've done for the past 20 years falls apart, I'll say, guess it's no. If everything I've done is based on a confounding variable I haven't measured, but for the moment, I mean, we're doing empirical research here. So empirical research means we think we have something that measures something, and we've tried to see how biased it is, and we think we're taking into account the biases, and for the moment, it holds up, but maybe it won't at a certain point in time. I don't know. It's not me, it's the birds that count. Okay.